ceremony. Please wait until I invite you to start with your presentation. Hereby I open this academic ceremony at Maastricht University. And first of all, I welcome Mrs. Juliane Hermann, and she will defend her academic thesis, MALDI mass spectrometric imaging methods for localization and identification of pathophysiological relevant regulators in tissue samples. And I will also welcome the members of the degree committee, and in particular, I welcome the three supervisors, Professor Leon Schurgers, he is Professor of Biochemistry of Vascular Calcification at Maastricht University, Professor Joachim Jankowski, who is Professor of Molecular Cardiovascular Disease at the University Hospital and the University of Aachen in Germany, and I welcome the third promoter, Professor Vera Jankowski, and she is Professor of Biochemistry at the University Hospital and also at the University of Aachen in Germany. And as you probably know, this will be a double degree of University Maastricht and University of Aachen. I will introduce the 5 opponents later during the ceremony. And of course, I welcome all the followers of the live stream. Welcome to you all. And Mrs. Hermann, may I invite you to give a summary of your thesis. Please go ahead. The floor is yours.
Thank you. So, dear Prorector, dear members of the Corona, and dear colleagues, friends, and family, I would like to thank you for your time to evaluate my thesis and for the opportunity to present my thesis today. The thesis was performed at the Institute for Molecular Cardiovascular Research in Aachen under the supervision of Vera Jankowski. And the topic of my thesis was the method development for mass spectrometry imaging and the application of these respective methods to different problems regarding cardiorenal diseases. So, cardiovascular diseases are the most frequent cause of death worldwide and are closely related to chronic kidney disease, which are often underestimated and recognized much too late. So that there is an association between chronic kidney disease and cardiovascular mortality is demonstrated in this graph. So the 24 to 34-year-old dialysis patients have the same risk of dying caused cardiovascular diseases like healthy persons between 75 and 84. So it's obvious that there's a strong increase for cardiovascular mortality due to chronic kidney disease. It is known that chronic kidney disease and cardiovascular disease are promote and influence each other, but the underlying molecular mechanisms are not fully understood. So there must be mediators which promote and support these interactions, but it's not clear which ones. In context of CKD, it's primarily uremic metabolites which accumulate in the plasma and influence the heart. For detection and identification of these unknown mediators, the mass spectrometry is suitable. For mass spectrometry identification, proteins are digested by enzymes like trypsin and resulting in a peptide mix. The molecular mass of these peptides is detected and illustrated on the first mass spectrum. The ions in these mass spectrum are selectively fragmented and analyzed by a second stage of mass spectrometry and resulting in the mass ion fragment spectrum, which represent the amino acid sequence of the peptide. And by using protein database check, it is verified that the experimental identified sequence and the theoretical sequence are identical. So there are indicators that these uremic metabolites, which accumulate in the plasma, promote the development of post-translational modifications. And in the last few years, the detection of these post-translational modifications in urine and plasma by using mass spectrometry approaches have increased. But what exactly are post-translational modifications and how they can transform native proteins into potential pathological mediators. So for this, I want to give you an example of albumin as the most abundant plasma protein. So there's on the one hand, the enzyme catalyzed post-translational modifications, and these occur in specific enzyme recognition site. And the most popular example for this are kinases, because kinases are known to promote or to regulate protein functions via post-translational phosphorylations. On the other hand, there are non-enzymatic post-translational modifications, and these are spontaneous reactions that occur that electrophilic metabolites, such isocyanate reducing sugar or oxygen radicals, coming through the environment of nucleophile amine side chain, the primary amines. So the impact of these post-translational modifications become more and more evident. And so I've written a review about this topic and to highlight the clinical relevance. But this review described also that the biggest challenge is the detection, the identification of these post-translational modifications. And through mass spectrometry, we are able to identify a various of different circulating substances in urine and plasma. But there's also an imaging method based on mass spectrometry, which we can use for identification of non-circulating substances, for example, in the tissue. One of the most popular mass spectrometry methods in field of imaging is the matrix-assisted laser desorption ionization time of light mass spectrometry, or MALDI-TOF, MALDI imaging. And for such approach, the analyte is mixed with a matrix substance and placed on a conductive surface. And the ionization is performed by using a laser beam, and this laser beam resolves out the molecules from this crystalline structure. And at the same time, the energy of the laser is absorbed by the matrix molecules and transferred to the analyte molecules. And these molecules are transferred in the mass analyzer by means of an electric field. And in this mass analyzer, the molecules are separated according to their molecular mass. So the small molecules move farther through the flying tube and reach the detector sooner than the large one. And the detector registered the molecules and transmitted this to a digital signal. To get spatial information of this, we lay a coordinate system over our tissue sample and define measuring points. And for each measuring point, we generate such mass spectrum. And finally, we have a representative mass spectrum for the whole measuring area. And now it's possible to choose a random mass signal, and on each measuring point, the intensity of this mass signal is displayed. And so we had an overview about the distribution from the intensity of this specific mass signal. So with mass spectrometry imaging, we can identify and localize mediators directly in the tissue sample without the using of antibodies and without staining. And the mediators we can detect in a wide mass range in one analytical step. And this offers the possibility of an association to histology, so adding the molecular level to the microscopic level. But there is a gap in the availability of standardized protocols for the sample preparation of MALDI imaging. And this leads me to the first part of my thesis, where I've been focused on the optimization and standardization of the sample preparation for the MALDI imaging. And for this, I chose various parameters from the literature and tested them systematically. And here I have focused on three main aspects: the enzymatical digest by trypsin, the washing steps before and after the antigen retrieval, and different coating parameters for the matrix. And all parameters are validated by the intensity of the molecular mass of the mass signal and the signal to noise ratio of the measurement. And at the end, the protocol was established, validated, and also transferred to other organs. That is demonstrated in the right part, so we can apply this sample preparation protocol for kidney sections and also for heart tissues. And this optimized sample preparation protocol was published in Analytical and Bioanalytical Chemistry and allows us to accumulate MALDI imaging data in a reproducible and comparable way. The images by MALDI imaging resulting only in two-dimensional illustrations, and this is very difficult to evaluate protein distributions in a whole organ when we have one single section, and also difficult to detect histological structures or specific cell types. And this leads me to the second part of my thesis, where I try to solve this limitation. And here we want to fuse different image modalities with MALDI imaging. So what we do here is we made serial sections of whole organ, and every 4 consecutive sections form one data set. So we have the first section for MALDI imaging measurement, a second one for, in this case, NHE staining, a third one for Gomori staining, and the last one for an immunohistochemical method. And now we fuse all these imaging data and want to reconstruct our organ. And as an example for such model, here is illustrate the reconstructed MALDI imaging data of a mice heart in green and the corresponding HE stained images of this mice heart. And this model we can zoom in and we can also zoom out for a better overview. We can rotate this model and we can cut this model from any angle. Unfortunately, it's not allowed to show movies in this presentation, so I hope this model give you an imagine how the model should work. And also we have the possibility to screen out the individual layers or to superimpose all the images we have. And this provides a link between the molecular level to the microscopic level. And this automatically fusion of the image modalities and the reconstruction at a 3-dimensional model was published in proteomics clinical application. To sum up my thesis, I have optimized and created a standardized protocol for the sample preparation of MALDI imaging, and we established a method for an automatical fusion of different image modalities and the reconstruction at a 3-dimensional model. And this helps to increase the acceptance of the method and provide the significant support to other projects. So the MALDI imaging could applicate in various consortia and support different projects. For example, IntriCare, Carisian, and also in some SFB projects. And the MALDI imaging and especially the 3-dimensional reconstruction are part and a central project in the SFB TRR 219. So this method will allow a deeper molecular understanding of the pathology of CKD-associated CVD and will help to detect and identify relevant mediators in these diseases. And this information could be used for clinical research or for the development of new therapeutical approaches. And regarding to different supported projects, so there are 3 first authorship publications, which are the main part of my thesis. And we have some successful support to other projects by using the MALDI imaging. And also some posters and oral presentations were made and found a large interested audience. And with that, I want to close my presentation. I want to thank the members of the Corona for your time and for your interest of the thesis. I want to thank my supervisors for all the support in the last years. And I want to thank my colleagues, my friends, and my family for the support in the background. So thank you very much, and I will now give the floor back to the pro-rector. Thank you.
Thank you very much, Mrs. Hermann, for your clear presentation. And we will now move to the opposition. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Erik Biesse. He is Professor of Experimental Vascular Pathology at Maastricht University, and he is also the Secretary of this degree committee. Professor Biesse.
Thank you, co-actor. Dear candidate, first of all, my congratulations with this thesis. It's excellent work, and read it with a lot of pleasure. And I would say also my congratulations stretch further to your supervision team, the hand of which I can recognize in this masterpiece. We have been collaborating in the past already through our PhD student, Valeria Saar, who has been working on PDMs and for who you did some mass spec imaging analysis. So my questions relate to chapter 3 to start off with. And chapter 3, you present a very elegant procedure for the 3D reconstruction of mass spec imaging images, aligning also some non-mass spec images like H&E and Komori stain with the mass spec imaging. And I think that's a very important step forward. You use for this a two-step registration procedure, and I think that is very appropriate because the mass spec imaging itself already leads to non-linear deformations of this issue. And often you like to integrate two non-identical tissue sections, subsequent tissue sections, which also requires more intelligent sort of alignment. So you use this two-step procedure where you use first a sort of linear registration step, followed by a non-linear elastic registration. For the latter, you often need to define landmarks. Did you do that yourself, or did you do that in an automatic manner?
So, yeah, highly esteemed opponent, thank you for the question. I think it's an interesting question. The algorithm behind this automatic registration have, you said correctly, two steps. So the first is to deformate the individual sections. So when we have our sectioning process, the organs have some deformations, what we don't see with our eyes. And the algorithm, compare these sections, and every individual sections would be to deform in the correct position and in the correct site that we have and exactly superimpose. I can say for kidney sections is quite easy to find the right position for the algorithm. For a heart, what it's nearly round, it's very difficult, but it's possible. But basically is that we have the sections nearly in a correct position and not a rotation of the section.
And I can understand that for kidney it's easier because you have this sort of this very clear structures, structures that are a bit irregular. But did you check each section because there may be some sections that have been deformed in a sort of excessive way that the algorithm by itself has difficulty in aligning? Did you need to give them a hand in defining landmarks yourself, or did you exclude these sections?
No, we include only the sections, and the algorithm worked absolutely automatically to find these points. We give to the computer the first section we have as a start point, and on this section all other sections what come later were deformed in this way or tool.
Very clear. I think so my next question is, what level of precision do you reach with the alignment? So did you take efforts? Because I couldn't trace that back in your thesis. So how precise was your alignment on average?
So how much?
Micrometer, is it 10 micrometers, 100 micrometer?
Okay.
What level of precision do you reach? Because that has also some repercussions in sort of linking the layers, if you like to do that in a subsequent study.
Yes, it's a good point. So for the theory sections, we need 5 micrometers for each sections, because it was very difficult to do it with a thinner sections. But this is a processing for the sectioning. So when we have one completely data set, we have a range of 20 micrometers. But we do also is that when one of these 4 sections are a defect or incomplete, then we removed all 4 sections from the model, that we have a better comparison of the different modalities. And at the end, we fuse for the kidney. I know it was nearly 100 sections. So, and for the myocyte in this part, it's nearly 40 sections. I know, because here is the problem in the limitation for the technical resources. So with a higher resolution to fuse all this imaging data, it's very, very hard. And so we have here the compromise to have more sections to to superimpose or to get a lower resolution. It's a little
how does the precision compare to the resolution of your mass spec imaging itself? Because I think that's there's no need to go to a higher resolution or higher precision than the absolute sort of resolution of your mass spec imaging, because that will be waste of effort, I would say.
Well, that's correct. Also, the size of the MALDI imaging datasets are nearly about 160 gigabytes for one image. And regarding to the microscopic part, what we have is, so we try to make the pictures in a very high resolution, but it's with the microscope not so easy to get a high resolution when we have an overview of the organ, or to zoom in with the microscope to reconstruct the microscopic image. So what we do at the moment in the following project, we want to integrate the data files of a slide scanner, so of the virtual microscope, to have a better or more quality or resolution. But these data files also are very, very high in data size, and so that we have once again the problem that the size on data are so much, and it's very hard to handle this with one algorithm. And so we have a lot to do to optimize this algorithm, that we can use it for a completely organ at the end.
Okay, so I have a brief question, if the pro-actor permits, on chapter 5. Nope. So then I give back the word to the pro-actor.
Thank you very much, Professor Biese. The opposition will be continued by Professor Vleuke. He is Professor of Nephrology at the University Hospital and the University of Aachen in Germany. Professor Vleuke.
So, dear candidates and dear promoters, wonderful work, which I know well from the thesis exam in Aachen already. Like Eric Biesen, I would like to focus a little on chapter 3. And we've been discussing this is there any way to do MALDI imaging of stained sections? I mean, your obvious problem is that you have to do serial sections. And if there are 5 micrometers minimum, it's one cell layer. A cell, at least the red cell I know is, I think, 4 micrometers. So why can't you do everything on one section? What is there a fundamental problem, or do you think you can develop this?
Dear highly esteemed opponent, thank you for the question. The problem here is that we have here limited a technical limitation, because the MALDI is using in the ionization process by using a laser, and the laser is light at the end, a beam of light, and this is, well, it could be small, but it could not infinity small. So we have here the limitation, and at the moment the best or the lowest laser size what we have and we can use is 5 micrometers, so that we here have a technical limitation of 5 micrometers. And we can go nearly to the cell resolution, but we can not so much deeper in the different cell types or cell compartments.
Why can't you pour some dye on the section before you do the laser? I mean, I'm surprised you can use paraformaldehyde fixed tissue. I always thought this denatures everything.
So we have here the choice to use paraffin material. We have also the choice to use a fresh frozen for our imaging. It's it's dependent on the question you have. Problem here is formalin fixed tissue is a standard material or sample material in the research on the clinic and overall, and fresh frozen is difficult for storing or for the storing place, and it's also a little bit difficult to sectioning. So the quality is an important point in an imaging method. So when we have a nice picture at the end or a nice image, it depends that we have a high quality section, and the quality of FFPE sections are much higher than for fresh frozen material. Regarding to this, we have here also the fact that fresh frozen material, it's not not even processable that we can section for 5 micrometers. So in some case, I know for liver maybe it's hard to get a sectioning of 5 micrometers, so that we here have an thicker sectioning of the tissue, and then we have to adjust our measurement features regarding to the number of laser shoots or the intensity of our laser.
Phrase my question. When you have a, let's say, 5 or 6 micrometer paraffin section, you pour some dye over it. You don't have an ideal stain, but you just pour dye over it. You take a picture and then you destroy it with your laser. Why doesn't this work?
Why we can create these images or of the proteins, you know? Well,
I mean, spatial resolution is very important for me in the kidney, where I'm not talking about a trivial organ like the heart, which is just a piece of muscle. I know that Michael Lerck is a little irritated here, but compared to the kidney, the heart is very simple in structure, whereas a glomerulus is extremely complex, and within a very low spatial confinement, you have very different cell types. So spatial resolution is essential for me, unless I ask about what is the type of amyloid that's deposited in the kidney, and that's where it's already in clinical application. But if I want to know specifically does the, let's say, endothelial cell have VEGF receptors?
Mhm.
I need to know where the endothelial cell is.
Okay. Yeah, that is exactly the reason why we want to combine the MALDI imaging with the histological part. So we can do it also for the two-dimensional images, that we have an overlay about the high MALDI resolution images and the histology, so that we have the comparison about the structure and the specific cell types you're looking for, and the mass thickness of protein which are in this area. And this is also the reason why we create the three-dimensional reconstruction, that we have a better understanding and yeah, better together with histology structures and the protein distribution. What we can also do is, when we have our MALDI imaging measurement and we have the corresponding or the same section for stain with and the method, and can see or detect the glomerulus part, in this case, we can extract this defined area from the whole measurement, so that we only have the spectra or the information in this area, and ignore the signals of the rest of the spectrum. And so we have a very specific focused, or a better spectrum to find the specific substances or mediators.
You're still saying you need an overlay. You cannot get around the one stain and one MALDI slide.
No, it's we try this, but it's not working in a good way. So we have the first only really for MALDI, and a second for histology. Or what we can also do is, when we do our MALDI measurement, and we can stain this measured tissue after the MALDI measurement. This is also what we can do. This is possible. It's working. But normally we don't do it in this way. We use corresponding sections.
But don't you have a Swiss cheese after MALDI measurements?
Is a what?
A Schweizer Käse.
This is a little bit depending of the resolution and of the thickness of the tissue. When we use 1 micrometer thick sections, then we do not have enough tissue for staining. But in case of 5 micrometers and a good resolution, but not the highest, it's also possible to stain it after. But normally we use this same section for do the identification, so the MS2 spectra. And in this case, then we have lags in our tissue, and then it's not possible to stain, or it's make to stain after.
Okay, thank you.
Thank you.
Thank you, Professor Vleuke. The opposition will be continued by Professor Leerke. He is Professor of Internal Medicine and Cardiology at the University Hospital and the University of Aachen in Germany. Professor Leerke.
Dear candidates, thank you very much for your very nice presentation and excellent work. Let me start with a technical question. If you do fixation of your specimen, are you in danger of changing the mass of the proteins by the fixation? This is to be taken in consideration.
Sure, dear highly esteemed opponent, and thank you for an important point. No, the reason is, for the fixed material, we use the enzymatic digas by trypsin. And so we have, before we start, we have our fragments of the protein. What is not possible for fixed material is to detect intact proteins of the cross links through the fixation. That is not possible. But the reason why we do the triptychal digest because of this problem that we have cross links of this fixation, and then we have a lot of single fragments, and our task is it to check which fragments could be to the specific protein to try to recombine this. So intact proteins can we only detect when we don't have a fixation material. So it's only possible to use a fresh frozen material without an fixation. Then it's also possible to detect proteins in a higher mass range, but with fixation material is not.
Okay, thank you very much. Another question. I'm always worried about specificity. So as I understand, as you get a mass of your fragments, what is the next step to validate your results? Meaning, if you do an interpretation and you detect a certain mass and you believe that this mass connects to this fragment and to that protein, and that's the interpretation which will show in the figure, how can you convince yourself this is really the case? So what is the second best method, if you believe your method is the best one, or what is the gold standard to control for your results?
For check our masses, no, I start another way. So when we have our samples, normally we have an idea for what proteins or also modifications we are looking for, and then we can we can look at the sequence of this protein, and when we have this sequence and we have our triptychal digest, we can calculate which fragments or which molecular fragments are possible present in our spectra, and then we choose these we try to recognize the signals of these masses what we calculate before, and when we found this signal, then we do a second mass spectrometry measurement. So we have our fragment spectrum, and with this fragment spectrum we can recheck if the amino acid sequence is corresponding to our fragment what we are looking for, or is it another sequence and not for the specific protein. And for this check we use the database, and the database give us a score or probability in which match the spectra are identical or not. And
can I ask you, in your opinion, what is the relevance of classical protein chemistry? Meaning, is there a requirement to validate spectra by using classical antibody-based methods, or is not required?
Can you repeat the question?
Sorry. Let's say classical protein chemistry, meaning Western blots, for example, which obviously wouldn't have the spatial resolution as the nice methods you've been showing us, but just in a general way of validating your mass spec analysis, is there any relevance of classical antibody-based protein chemistry?
So the method like Western blot we use for a mass spectrometric approach without the imaging compound to separate or to homogeneous our material, but it's a little bit difficult to the imaging part, because in the imaging part we can now separate before we detect all molecules are in these sections are present, but we validate our results with the comparison to the multi-imaging spectra and results to the high resolution mass spectrometry. And when we can analyze the same signals or the same modification, have the same results with the coverage of the database spectra, then yeah, I think it is a good way.
Can I have your opinion about the future? Let's say this pandemic was in 10 or 20 years from now. All the tests, all the antibody tests we see, or antigen tests we see at the moment, are all antibody-based. Do you believe in the future this will not be the case anymore and everything is going to be by mass spec?
So that's an important point. No, I don't would say that mass spectrometry is the perfect method and is better as every method what we have at the moment. I think mass spectrometry can support in a good way, and in the clinical routine at the moment, a lot of mass spectrometry based methods are working, and it's also very difficult to bring the methods what we used here in the research in the clinic, because we have a non-targeted approach. So in this case, we need high much support for IA algorithm to help to analyze these spectra, because when you don't know what you are looking for, it's hard to establish this method in a routine, because it's too long. And I think in this case for the pandemic, corona antigen test is very, very faster, like an analysis with the mass spectrometry. And so far, no. But I think it's a good support, or it would be a good support in the next years.
Thank you very much. And I give it back to the pro-rector.
Thank you, Professor Leerke. The opposition will be continued by Dr. Schanstra. He is Associate Professor of Nephrology and also Research Director at the Research Institute INSERM in Toulouse in France. Dr. Schanstra.
Thank you very much, Mr. Pro-Rector. Dear candidate, I would like to echo the words of the other opponents by thanking you for the very nice presentation and the book. It's a pleasure to read. And also would like to congratulate the promoters. I have a few very technical questions. So from chapter 2, where you did a lot of optimization work to try to make it work. You concluded that Sigma, so the company, provided the best trypsin solution. So did you check different batches?
The highly esteemed opponent, and thank you for this comment. We could say that we in our tests we have the best results with the trypsin of Sigma. We tested also different batches, but only 2, and there are no difference between these batches. But it's it's a part. It's not in the publication, but we tested it for 2 different batches of Sigma in this case.
Could this become a problem with time? I mean, would you like to implement such an approach in research in the long term, comparing samples in the long term? In 10 years, you have other trypsin. So could this be an issue?
But so we use Sigma at the moment until 5 years, and until now we can't see strong variations in the quality of the fragmentation, and so far I would say it's very good.
Okay, thank you. And so those were all paraffin embedded sections that you used. So those were freshly embedded paraffin sections, or there were already samples that have been around in the lab for a while. I'm asking this out of curiosity because of the use of historical samples that were taken, human samples, very rare human samples were taken 20 years, for example, ago.
So
did you?
Yeah, it's an interesting question. The samples which I'm working with are very freshly and so far from running my studies, not older as 1 or 2 years. but I know that in the literature is also described that the MALDI imaging could successfully use to paraffin samples which are older than 24 or 25 years. So I think it's quite possible. What I can say is that the time of the samples or the quality of prepared samples for MALDI imaging, when I have the matrix on the slide, then we have a loss of quality after a half year. So this is
with the matrix. Okay.
Yeah, this
with
is
the matrix.
I can say from my work. Yeah, and I think it's possible for samples nearly 25 years, but I have no idea why we can not use this method to older material.
Okay, but it could be potentially, you could potentially use those older samples. Okay, thank you for those answers already. I have another question related to coming back to the resolution. So would there be an approach to improve the resolution by focusing on specific proteins in specific, in the kidney, in specific cells, for example, podocin to identify the podocytes, transporters to have the tubular, to identify specific tubular segments. So could you recalibrate your sample by focusing or trying to find specific proteins and to improve the resolution, to go beyond these 5 micrometers, which you use technically?
Yes, so what we do, or what we can do is when we have our image result, and yeah, we have it in a high resolution way, we can extract different areas of this picture, of this measurement. We can extract the information only from this specific area, and then we can check it in a separate way. But for this we have the help by using histologic stained sections behind our mass spectrometric tissue.
But you could not, as was indicated by Professor Lierke. So if you say, okay, I can identify specific proteins, you said we can focus on specific mouse. So once you have identified your specific fragment, can you do not completely screen your slide and say, okay, this is potentially podocyte because this fragment is from podocin? And this will help you to delineate cells more specifically. Or is this completely bad idea? That's also possible.
Don't say it's a complete bad idea, but
thank you.
I don't think about this approach, but yeah, we can discuss about this, but at the moment we do it in a very other way.
Okay, thank you. So talking about the competition, so spatial transcriptomics is getting stronger and stronger. So would this be a competitor or would this be a helper for you?
That's an interesting point. At first I would say it's a helper, because so spatial transcriptomics is focused on detection of mRNA or DNA. And this is not the topic of MALDI imaging. We can also do it with the method, but normally the focus is more for lipids, metabolites, proteins, and so on. And when the spatial transcriptomics, and when I remember correctly, it's nearly the same resolution what we have at the moment, then I think it would be nice to compare both methods. On the one hand, we have the mRNA information, and on the other hand, we have the protein information, what is the result of this in the cells. So
okay, yeah, that was one of my things. So you believe they are at the same resolution currently as you are?
Yeah, I think so. I'm not sure, but I think so. It's nearly 10 micrometers.
Okay. I have another question on chapter 4. It's not about the signaling of NOTCH3, but it's about the mass coverage of your YB1 protein. So did you did you were able to see the to sequence the entire protein and see all the modifications, or was only parts of it? So I have no idea about the size of this protein. So other granulation sites, for example.
Yeah. So for the coverage, I can say when we check, when we do our check with the protein database, we get a sequence coverage. So how percent of this what we detect is coverage to the theoretical sequence. And when I, auch in this case, remember correctly, we are to 42 or 45%. In this work, we had also found other modifications in this protein, but in this case, it was not the, or had no impact on this study, so that the focus was on this one special fragment with the double gonadulation. But there are also other, but we have no more studies to these fragments.
Okay, so thank you very much for the nice answer, and we'll give back the word to the prorector.
Thank you, Dr. Schamstra. The next opponent is Dr. Scheijen. He is Assistant Professor of General Internal Medicine at Maastricht University. Professor Scheijen.
Thank you, dear Pro-Rector. Dear candidate, I also want to congratulate you with this nice work. I read it with great interest. And for me, it's really a lesson in MALDI MS imaging, so it was really nice to read. I also want to include your promoting team. And I also want to thank Professor Curtis for inviting me here today. What I also liked was your 10th proposition. The problem is not the problem, but the problem is your attitude about the problem, from Jack Sparrow. It's a pity that Johnny Depp is in court now, but it's a really good proposition. My compliments for that. But okay, I'm also here for a sort of academic discussion. And I want to start off with some questions about chapter 2. You are optimizing this sample preparation, and you're looking at a really long list of parameters. And in the end, you're using a signal-to-noise ratio of specific masses as a sort of outcome parameter. Just for me to understand, based on what criteria you selected these masses?
Dear highly esteemed opponent, thank you for this question. Yes, so the problem was that we don't have a target molecular protein for these optimizations. So we check every parameter and are looking for the best or for the highest signals what we found, and the signals what represent a nice image or a good distribution in our organ. And this is the reason why it's a little varies in the different parts. But here we try to focus on the signals, and because we have no target protein, we do it for the whole measurements. We use a random signal and to try to evaluate our results.
I see in the text indeed that you mentioned several masses, for instance, a mass of 8A2, and then you describe this amino acid sequence, and then you say which may originate from beta defensin, for example. But it sounds to me which may originate that you are not really certain about this amino acid sequence. Is there some uncertainty about it, or did I understand it correctly?
So no, that's what. So we choose signals, and at the end we identify these signals what we use for our validation. But this has had not a higher aim why we do it for this sequence. It was only to identify the signal what we choose for our.
Well, the reason why I am interested in this is, of course, because the removal of paraffin is a crucial step. You know, formaldehyde can react with basic amino acids such as arginine and lysine, and then can form methyl bridges and form inter and intramolecular protein cross-linking of protein. I mean, can you say something about the formation of this of these crosslinks? I mean, would it not be better, besides looking at signal-to-noise ratio, also to look at some modifications because of the formaldehyde preparation of the sample? I mean, can you say something about the quality of the sample based on several masses you see in comparison to a fresh frozen tissue?
Thank you for the question. It's an interesting point. No, regarding to the intensity, I can't see difference, because between fresh frozen and the paraffin fixed, the only difference is that we have for fresh frozen more options to detect other substance classes, because lipids, for example, are also included in fresh frozen materials. So we have a lot of more signals as in paraffin, for example. But the intensity or the quality of the intensity is is not very different. So only more signals.
And Professor Schanstra was also talking about this brand Sigma Aldrich of trypsin, and you also tested the two other brands, Roche and Promega. I was really surprised that there was that big of a difference in signal-to-noise ratio between these brands. You were also surprised, or?
Yeah, I was surprised in when I remember correctly for Sigma and for Roche. Promega was more the problem that Promega come with a resuspense solution for this trypsin, and I think that this special solution for the trypsin in this case, what the big difference between. But I also was surprised that the manufacturer of trypsin could so different.
Yeah, and that there's a big difference in purity or something like that.
Yeah, something like that.
Okay. Then I have some questions about chapter 5. I mean, in chapter 5, you gave a really nice overview of the status quo of MS applications as a diagnostic tool. And you published it in a really nice high impact journal, impact factor 14.2. I looked it up, really nice, congratulations with that. And I was really intrigued by the different approaches. You described different approaches of the PTMs, analyzing the PTMs. And I'm working at the moment also at some modification of insulin in a mouse model of diabetes. And a very small molecule called methyl is modifying the insulin, and you get a mass shift of around 54. I'm really struggling how to detect and what kind of approach I need to take for seeing this modification. Can you give me some advice about that?
It's really nice to ask me that. So when insulin is a very small protein, or
it's around 6,000.
Yeah, this is a good size. So in this case, I would say it's not a problem to detect it with MALDI. And for I think insofar you are looking for glycosylation.
Yeah, you can say that. Yes.
So for this, from my experience, I can say I never detect glycolizations until now. But I know that in our institute or in our working group, it's possible to detect the glycolizations. It's also only possible to choose other matrix for a better detection. But with that protein size and a possible glycolation, I don't know why we can't detect these modifications. I think it's really possible.
Yeah. At the moment, I'm just taking the pancreatic tissue and I crush it, and then I isolate it with immunoprecipitation. I isolate the insulin. And do you also think that you would be able to detect this modification really in the intact tissue, like in an FFPE tissue? Do you think that is possible? This
Yeah.
small of modification?
Yes, yes, it's possible to detect it in the paraffin material. What I don't can say before is dependent of the amino acid sequence of the peptide, because trypsin has.
I'm quite sure, Mrs. Hermann, that you have noticed that the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and the quality of your defense, and please await our return with the results of our deliberation. I see that you are all back. Mrs. Hermann, the degree committee here present online has assessed the quality of your thesis and the quality of your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And this degree is granted you from Maastricht University as well as the University of Aachen. It's a double degree. Professor Schurgens is authorized to confer upon you this academic distinction in accordance with Dutch and German university custom and law. And I invite your supervisors now to take the floor. Professor Schurgens.
Thank you, Mr. Prorector. Dear candidate, dear Juliana, I first ask you about scientific integrity. So I will ask you a question. And if you agree, you just say, yes, I promise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Juliane Hermann, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university, shown by the beadle.
Thank you.
Juliane, dear Dr. Hermann, congratulations. The Laudatio will be given by Professor Vera Jankowski. She knows you the best, so I'm pretty sure she knows how to acknowledge your achievement of today.
Thank you.
Dear Professor Jankowski, the floor is yours.
Thank you, Leon. Dear Juliane, it is a special honor for me to give you the laudations for today, as you have successfully completed your doctorate. Let us think back 7 years. Dear Juliane, you have been working as a research associate in our Institute for Molecular Cardiovascular Research since January 2015. Your scientific focus you have found in the methods of imaging of the reflex mass spectrometer. You started your study in the toxicology at the Charité in Berlin. In this study program was also my lecture, Chromatography and Mass Spectrometry, which you heard. This lecture made you want to get to know mass spectrometry better, so you came to our institute. The first thing you did was a master thesis for the toxicology at the Charité Berlin. This master thesis was written and defended by you with the title Establishment and Validation of an Imaging Mass Spectrometric Methods in the Context of Toxicological and Cardiovascular Questions. This master thesis was graded very good at the Charité in Berlin. When you followed us, or especially say the mass spectra to Aachen in 2016 and have now completed your PhD 5 years later with a rarely awarded predicate summa cum laude. You have all well earned this special high destination in the 5 years you have been here in Aachen. You accepted Aachen as your new home and also started a family. Your husband, Sebastian, moved to Aachen with you, and your children, Mika and Flynn, were born in Aachen and now at home in Geilenkirchen. Also during the time you wrote new methods for sample preparation using the mass spectrometer ultraflex 3 imaging, and thus produced your first publication in analytic bioanalytical chemistry. You was always present in your presentation. The collaboration in the SFB S03 was left you little time to exploit the full potential of the mass spectrometer. However, you succeeded in some co-authorships, which you also worked on with a lot of scientific dedications. For example, you screened the CXCR6 in the liver of the medical clinic 3. This work and the figures have subsequently been published in Gastroenterological 2019. For the SFB, you detect modified peptides in ApoC3 in kidney tissues. This work was published by Jason. The interest in your work with the Rapiflex was grown with many collaborations. So you proposed the question of whether guanidulated YB1 can also be detected in kidney tissues with MALDI imaging, and was published in Kidney International 2020 after the work was completed. Likewise, carbamylated sortilin was detected in tissues and published in Kidney International 2022. To the numbers of co-authorships that have brought the SFBS03, you have followed your own research. To the second own publication with the question of the 3D model was followed with a high interest. The work has been published in Proteomics and has just been reported by you in your talk. Besides the manuscript and the dissertation, further collaborations have already been started. For example, with the University Maastricht, with the Department of Pathology and CARIM, the modification in atherosclerotic plaque tissues are being followed up with very great interest, and carbamylation is being detected. Similarly, collagen modification are being investigated with the Aachener pathologist, and the cardiologist from Hamburg has also some questions that still need to be investigated soon. I could expand the list considerable now, which I will not do now. Many cooperation partners, and I can only confirm the award with the highest prize. Without paying attention to the times and the days of the week, the tasks are successfully completed. Not only that, but you are also already to be very in a love and a good mood. This shows how gladly you make this work with the Mars spectrometer. Often do the guidance of your family. Finally, I would like to congratulate Dr. Juliane Hermann on this brilliant graduations and hope for further years of successful cooperation and great results in the Institute of Molecular Cardiovascular Research. Congratulations, Juliane. I'm very proud of you.
Schön.
Thank you.
Thank you, Professor Jankowski. Esteemed Dr. Hermann, dear Juliane, it's my pleasure to congratulate you with your double doctorate and the honor that you have acquired today. And I do that also on behalf of the Board of Deans of Maastricht University and of Aachen University as well. And I would like to share some impressions of the committee with you. We have enjoyed your clear and excellent presentation. And with respect to your thesis, we have seen a nice and easy to read thesis. And also we have seen that most of the chapters are published in good quality scientific journals. In your defense, we have heard a proper defense. And we have also seen that although you did not answer all the questions exactly as we expected, we have seen and we are very satisfied with the quality of your responses, and we like the discussion. And also you have shown that you are very knowledgeable in the field of your research. And I would like to congratulate you, and not only you with this result, but also your 3 supervisors. Professor Leon Schurgers, Professor Joachim Jankowski, and Professor Vera Jankowski. Congratulations with it, fantastic result that we have seen today in the form of a thesis and a very good defense. And of course, I would like to include in my congratulations your parents, your husband, and also, of course, other members of your family and your friends and of course, colleagues and those people who have contributed in any way to the realization of your publications and of your thesis. And I wish you a very successful professional career and a happy personal life. And before I close this ceremony, I would like to thank all the members of both committees, of the Thesis Assessment Committee, as well as this degree committee. And in particular, I would like to thank all opponents for their questions and the interesting discussions. Maastricht University and University of Aachen appreciate your contributions. Hereby, I close this academic